Great, thank you very much. Alicia, while we're just waiting for questions to come online, would you like to start asking questions in the room? Yes, thanks, Rachel.
Question for Ed. Well, firstly, a comment, because I've seen your project grow and the amount of work that has gone into this is just phenomenal, so congratulations. But a question that patients may have about exogenous retinoids and taking vitamin A supplements, and people may be cautious about dietary implications of your work, so I'd like maybe you could comment on that.
Yeah, thank you. No, that's very, very fair. It's not actually something, as a basic scientist, I often consider, and we really are just looking at, so we haven't considered dietary retinoids. I don't know how they would affect it. I think we're too early in the project to really have an answer for that. I wouldn't suggest anyone go away and change their diet or their approach to their own self-care based on what I've shown today on retinoids. No, I think it's too early to say. I think we really are thinking about this from a basic science perspective as a fundamental pathway. I don't think the answer is necessarily going to be treat with retinoids or inhibit with...
But people shouldn't be concerned about...
I don't think there's enough evidence to say that, no, at all. Thank you.
I'm very interested in the DNA methylation patterns that you get in different cancers, and I'm wondering whether the two animal models have looked at the DNA methylation patterns in the pathways that they think are important in tumor initiation. So that's my first question. My second question is, we've invented 350,000 new chemicals in the 50 years since I qualified as a doctor, and I believe that many of these chemicals act epigenetically. So is there any evidence that the DNA methylation, that the DNA methylation pattern in cholangiocarcinoma can be affected by any of these 350,000 chemicals, which nobody looks at?
That's a very interesting question, and I think we know DNA methylation changes with age, environmental factors, there's so many things that feed into it. The work that we've done, so we look at the whole genome, and then we filter it down to about 2,500 300 base pair windows. So we are actually disregarding a lot of the genome when we're looking at the tumour signal here. The data's there, it's just that, I guess the time and where to start looking, because it really is a needle in a haystack in terms of what signals you're trying to pull out. But yeah, if you had the time and a hypothesis, you could use the data to answer that question.
So if I may. I have a question for you, Jen, actually. I think I probably have about 100 questions for you, but I'll try and control myself. What you mentioned about the cytokeratin and SOX9 Cres and the differences between them is really, really interesting. I know you've just moved into cholangio. So, do you think with animal models we need to be slightly more finessed about what Cres we're using? Because we seem to get broad brush recombination. We don't really get much information on cell of origin from that.
Yeah. Yes, we do. So I mean that's why we made. Is this on?
Yeah.
That's why we made accidental models, because we were trying to finesse our pancreatic models, which do suffer because we're probably not targeting the right cell of origin.
Yeah, I do think the cell of origin thing is really fascinating, and when you see those stark differences between those two promoter systems, it kind of highlightes that, but we don't think about that.
Histologically, you're looking at the RFP on there, and it looks like the same cells, before any tumour formation. So,
absolutely.
there are fundamental differences.
And Tim,
As a quick comment and then question for Alicia Marie. Your British Journal of Cancer paper, the 2022 paper, is one of my favourite papers. It's sort of like confirmation bias because I hate cups, so it's fantastic. My question is, in cup, does it matter what label we put on it? If we sequence them and find there's a vulnerability, they've got FGFR2 fusion, treat that. If they've got RDH1, treat that. And the label is fine, but the weak spot is.
Yeah, so that's exactly what the Capisco trial was trying to do. It didn't matter what, it was tumour agnostic. I think the results were positive from Capisco, so if you did molecular guided therapy, then you did improve outcomes. However, the proportion of patients with a targeted mutation was tiny. Most patients ended up on immunotherapy. And the big hitters that did well were FGFR2 fusions and BRAF V600E. So as we know, targeting these single mutations sometimes is really effective if that's the main driver, but it's almost always more complex than that. And I think with combination therapies, so for BRAF, for example, in colorectal, you use a different combination versus melanoma. I think actually I'm all for more information is better.
Yeah, I just wanted to follow that up with a comment. I've also toyed with this idea. Actually, does it matter what you call it if you find the right driving alteration? But I think what we feel is that the patient needs to be under the right team managing the disease. And so that gives you access to local regional therapies, expertise, clinical trials. So whilst it is important to get a diagnosis thing, I think it's much more important that patients are under a specialist team. And so being able to get that diagnosis then aligns the patient to those teams, the experts who can then look at some of those additional kind of factors of managing the disease. So it's just a comment.
I was just going to add on to that as well. I think sometimes there are also licensing issues for the drugs. There are a lot of drugs that, for example, might be licensed in the US or elsewhere as a pan cancer for that specific mutation, but in the UK only licensed if you have a formal diagnosis of that tumour type, which can make it difficult to access.
Yeah, and we're definitely sort of seeing that in CUP MDTs, where if you do have molecular profiling, you're like, oh, you'll get access to this therapy if we label you as, but that's not necessarily the right way to go about it.
Actually,
I was just going to make a comment there. Wouldn't it be better to get NICE to change their stupid thing? I mean, they are doctors, and we ought to be able to argue with them, because if you've got a mutation that's responsive to a drug, you ought to be able to access it whatever your cancer.
it would be nice not to have to randomly label cups as something, but that's a long story. I think we could be here all day talking about that. Actually, Alicia Marie, I just had a quick question about the CUPID algorithm.
Yes.
I know it's very, hopefully going to be very useful diagnostically, but is it sensitive enough to pick up these sort of potentially targetable mutations, or is it purely as just a diagnostic tool?
Yeah, so we don't do a high-depth sequencing to pick up mutations. We could build it into the assay, but we're trying to think translationally about how you get the test into the clinic, and I guess the more complicated and technological it is with higher depth of sequencing, the more expensive it is. So it's a trade-off between sensitivity and actually will we ever get it into the clinic. Cool.
Yeah. Hi, sorry. Just to the point about having to label as cholangiocarcinoma to be able to access these treatments. I just wanted to put in my 2 cents from spent about 1.5 years treating CUP in Addenbrooke's, where our pathologists were very keen to do whole genome sequencing on CUP patients. So we started actually picking them up from when they were first referred, and their first biopsy was done on fresh tissue because we had an extremely motivated CNS who made it all happen. And what we actually found, I mean, on preliminary small numbers, because they had to be fit enough to have treatment, but in about 2/3 of patients, we found a potentially actionable mutation that could be a referral to trials. And we also managed to repatriate people, that where the HPB MDT would not have accepted them as a cholangio, but because we had, a genomics MDT that looked at it and said, this looks like cholangio, they got repatriated and they ended up accessing immunotherapy and having access to these targeted treatments. All patients with cancer of unknown origin are eligible to have whole genome sequencing. This is funded centrally. I think they have put a caveat on it now that the patient has to be fit enough to have treatment, and you have to have, sort of treatments in mind. But it's just something to bear in mind, given that as you've shown, such a high proportion will turn out to have cholangiocarcinoma. Sorry, that was more of an annoying conference comment rather than a question. Thank you.
No, thank you. It's amazing that you can get access to it, because at the Christie Hospital, we were doing whole genome sequencing, but the infrastructure, we've just lost the funding and the infrastructure to actually deliver it. And some patients don't want to have the repeat. It's usually a repeat biopsy by the time they've come to us. But yeah, I definitely think that there's something more than where pathology fails and also radiology fails for cholangiocarcinoma. I think there's another group of cholangiocarcinomas that we maybe will only pick up molecularly, but it's just getting enough data to try and prove that.
Every hospital needs a Helen.
So if I can, I'm very much interested in the mouse models, so apologies if I'm kind of directing the questions towards you, Jen, essentially. But I was just considering, and this is something I consider in my own work a lot, the mutations that we choose in these models, right? And the main one that we've both used there, and to some extent, is P10. And P10 mutations aren't particularly common compared to, obviously, PI3 kinase catalytic mutations. Do you have any thoughts on whether we need to be a little bit more precise about what mutations we use? For P53 as well, and things like that. Sorry, it's a horrible question.
It's a thing that interests me a lot in mouse cancer, because even in the kind of Sleeping Beauty kind of trials, P10 deletion always comes out as like number 2 with if KRAS is the initiating mutation in just about every cancer type, and then it's not there in the humans. So is it maybe actually that we're missing a lot of the P10 because it's just potentially down-regulated rather than lost or mutated?
Absolutely.
So that's one question. We do have PIK3CA mutant mice, but since we were supposed to be working on pancreas, I had to rewrite the license to actually make it acceptable to continue. So yeah, we might look into that in the future.
Do you think you get the same phenotypes? Or
Yeah, you get different phenotypes in colorectal mice. I know that from PI3 kinase versus P10. Yeah,
that's interesting.
I'm not very keen on mouse models, but your mouse models seem to be showing us that there's some new drug from AstraZeneca. Can you tell us a bit more about that drug and whether it's going to be trialled or whether there's any phase 1 studies going on, or are you got to be buttoned about it? Because it did look like it was going to be rather interesting.
That's a long story because
they've abandoned it. Don't tell me.
The dual mTOR inhibitor they kind of abandoned because it wasn't a good mTOR1 inhibitor, and we always argued that mTOR2, particularly actually in pancreas, was more important, so that was quite irritating. But I do think, so we see as good responses with the KRAS mutant inhibitors and the pan-KRAS inhibitors now, and there are a lot of them in trial in other diseases, and phase 1's going on everywhere, so I'm optimistic. Maybe some of my oncology colleagues, I mean, I'm a mouse oncologist, so, but maybe some of my human oncologists colleagues can answer whether the KRAS inhibitors are going to get to cholangio patients anytime soon, at least the ones with KRAS mutations or activation of the pathway.
Okay, so the first one that looked exciting has been abandoned. Is that the case? I mean, I did some research on something that worked in liver cancer like about 30 years ago, and they abandoned that, and we showed in mouse models that it worked. And it's just very frustrating that you do a lot of work that works in mouse models, and then the companies don't take them to human development. So, and that was an economic decision by ICI. It wasn't anything to do with the data that we generated about a drug that seemed to work. So that's a bit why I'm anti-mouse models, because, you know, I spent a lot of time on it actually a long time ago on human xenograft models, but it's usually economic decisions rather than or side effect decisions that seem to drive it. You know, it looked a lot better than the standard treatment that everyone's getting at the moment, and yet it's been abandoned. So we ought to get on to AstraZeneca and say, why have you abandoned it?
Yes, it is frustrating, and I think the GEM models are expensive and time consuming, and sometimes data come from some of the less, or the easier to treat models, or the less robust models, and then decisions get made on that basis, which is frustrating. And a lot of people I hear everywhere I go that mice are easy to treat. I've never cured a mouse with pancreatic cancer. I mean, I've met 10-year survivors, patients, but I've never cured a mouse in 20 years. So, and out of over 100 drugs and combinations, I've seen maybe 3 result in regressions and sustained responses like that. So 3 out of 100. So yeah, when we can't take those forward, that's frustrating.
So I think I have quite a naive question for you, Dr. Conway. So you meant, and this might be misunderstanding, so call me out if it's my misunderstanding, but you mentioned that you're going to try and kind of make this slightly more precise for cholangio and improve it in that way. Is there any potential in the future that you might be able to differentiate subtypes of cholangio, get more molecular information from your approach and the methylation? Have you got one?
Yeah, no, I think we can, especially because of the different mutational phenotypes, probably change the epigenetics as well, so especially the IDH1 and 2 mutations. I think the key to it is having really good, robust training data that has all the clinical annotations, so that you know the outcomes and also the mutation profile. So we are trying to build our own training data and make sure that a pathologist has reviewed everything that we're then putting into the algorithm, because we found just from the TCGA data, some of the pancreas data was, not very good tumour fraction, not very good tumour content. It maybe wasn't pancreas cancer, and so we've excluded a lot from that training data and improved the algorithm. So it just goes to show that, it's what you put in is what you get out, and we are trying to curate it to make it cleaner. But I think we probably can if we have the right training data.
Yeah, nice. Okay, so maybe a slightly antagonistic question. Can you train your data from mouse models at all?
Well, so that's interesting. So in small cell lung cancer, we've used cell lines to train the algorithm, and then subtype small cell lung cancer. So yes, probably.
Oh, cool.
Yeah, that would definitely be something.
Not the answer I expected.
Yeah.
Great.
Well, it worked in small cell. I don't know.
Kevin?
Hello. I've got a question for Ed, or a comment for Ed, and then a question. So you said it's a poised response, but it's not a poised transcriptional response, because you don't see a change in the RXR gene sets. Therefore, I would argue that your hypothesis would be that it's not a transcriptional response. Therefore, your mouse experiment, you have proved that your hypothesis is correct. So your actual outcome for your mouse experiment is what you would expect if there was not a transcriptional response. So the question then is, so that's good, always cheer up. So you have to cheer up about that. And then the second thing is, so what do you think those cytoplasmic enzymes are doing, or proteins are doing, that is not to do with transcriptional signaling, but is maybe a secondary function of the retinoic acid signal?
Yeah, really good question. I'll clarify by saying I'm not sure we've been able to find the right gene signatures to confidently say that transcriptional response to canonical retinoic acid signaling isn't activated. We haven't been able to confidently show that it has been activated. So hopefully the luciferase approach will either show that it's not activated at all, or show that it is activated. In response to what else might be going on if this isn't happening through that canonical pathway. At the moment, we're really interested in how transretinoic acid is directed within the cell, and there's some literature out there that suggests that the balance of certain proteins can drive retinoic acid as a ligand away from the canonical receptors towards things like PPAR receptors, and that has a completely different promoter region and transcription factor binding site, and a completely different set of transcriptional responses. So if we do find from our luciferase that there's clearly not a transcriptional response canonically, there are other transcription factors that might be activated still by retinoic acid as a ligand. But we don't know yet. We're still trying to kind of pick it apart.
But I guess the obvious thing to do is to target those enzymes and those proteins in
Yeah. The reason we've done it the way we have with these agonists and antagonists for canonical retinoic acid is because those molecules are well defined and have been shown to work, and it was a simple way to get an initial answer to that question. But yeah, as I said in my talk, it's instantly become apparent that it's more complicated than that, and we can start to find more refined ways of, or start inhibiting different parts of that pathway and see if we can pick it apart. But I'm confident we can make some progress on that and understand it better. But it's fascinating but complicated is the short answer.
Wonderful. I think that's... Yeah, I think we'll call it a day. Thank you, everyone, for your insightful questions. Thank you very much to Ed and Jen and Alicia Muir for some wonderful talks. And I think we'll break for a coffee break and come back at 11 o'clock.
Thank you.